Hello, everyone. My name is Ivo Djidrovski. I am ESR number 9. And as part of the N3 project, my goal was to develop an iPSC airway model and apply it for toxicology testing. The hosts of my PhD were Dr. Mike Nichols, the CEO of Newcells, and Professor Lyle Armstrong from Newcastle University, who was my mentor for the past 3 years and is also the CSO of Newcells Biotech. I did my PhD at the Center for Life, which is located at Newcastle upon Tyne at the north of UK. So first of all, I'll give a brief introduction on the lung. And the lungs are a very beautiful and yet complex organ. They are divided into two main proportions: the conducting proportion, which is made of trachea, bronchi, and bronchioles, which filters the air before it reaches through the respiratory proportion where the air exchange is happening, and that is the alveoli. When you compare the cell compositions in these regions, they're different. In the airway, we find the goblet cells, which produce the mucus, the ciliated cells, which move the mucus around. We have basal cells and club cells that are progenitors. And we have neuroendocrine cells, which detect hypoxia, for example, and other functions. And on the other hand, we have the alveolar epithelium, which is composed mainly of alveolar type 1 cells that are very thin and elongated in direct proximity to the vasculature, allowing a smooth air exchange to take place between the lumen and the vasculature, the blood in the vasculature. When I started my PhD, there were two iPSC models, the two main ones that were available. One was in 2D, and the other one was a lung branching spheroid. Both were from the same lab, and I tried both of them, and when I saw the beautiful branching organoids, it was very exciting. But then I learned about the primary models at an air-liquid interface, which formed a pseudostratified airway epithelium super similar to in vivo-like structures. Well, this became slightly less exciting. And we decided to make something in between, isolate basal cells from the iPSCs and make something beautiful like the primary models. So I started off by differentiating 82 iPSCs during 14 days into lung progenitors using the previously described methods. And from this population of mixed progenitors, we isolated basal cells by using primary bronchiolar media, BGM, ROCK inhibitor, and irradiated 3T3 cells that will give growth factors to the basal cells and stimulate their auto-renewal. This has been shown to work with primary cells in the first place, which allows to expand them for multiple passages. And we use this method to isolate iPSC basal cells, which were also able to passage for multiple passages, up to 7 passages, I would say, generating huge amounts of cells, making it very attractive for high throughput, for example. But first, we have to make sure that these cells are actually similar to the primary cells. And for that, we did a quick immunostaining with basal markers such as NGFR, delta NP63, keratin 14, and integrin alpha 6. And as you can see, when compared to primary basal cells, both cells, primary and iPSC, are positive for all of them, which was very encouraging. And we straight away decided to put them on inserts. So these thinserts actually have a porous PET membrane, which allows 2 compartments to take place within the well. What we do is we plate the basal cells on the apical side, and once it reaches confluency, we remove the media from the apical side, inducing an air-liquid interface differentiation. And then we feed pneumocoat media, and at day 10, we supplement with a little bit of DAPT to enhance ciliogenesis, and we obtain a beautiful model which has ciliated mucus-producing cells, intermediary and basal. And this is how it looks like. As you can see, first it looks a bit messy, and then after 10 days, tight junctions can already be seen. After 30 days, these tight junctions are even more visible, and there's also a little bit of mucus here. And if you don't wash this mucus away after 30 days, it creates mucus tornadoes due to the movement of the ciliated cells. But if you wash it with a little bit of PBS, you can see a monolayer on the apical surface. The TR increases as it ages. After 30 days, it reaches around 300, which is very similar to primary models. And the H&E section also revealed some ciliated cells on the apical side, which were also visible on bright field. But first, I had to section to check if we have all the cell types inside this lung construct. So I stained for synaptophysin, which is a marker of neuroendocrine cells, mucin-1, marker of secretory cells, club cell protein 10, which is a club cell marker, and finally delta P63, which is the basal marker. And as you can see, it's only here on the basal side. And as the cells differentiate towards the apical side, we lose this expression. Then I stained for acetylated tubulin to reveal the ciliated cells on the apical side. And here they're together with Z01, which shows the tight junctions. I also did a secondment at Vrije University in Amsterdam, where I could collaborate with Jelle van der Boer, which helped me do a functional analysis of the ciliated cells and compare them to primary ciliated cells, to a primary model. And we saw that they both beat at 15 Hertz, which is the same as in vivo, and it's quite exciting. Here's what they look like. I'm not sure if they will be visible on the video. It may be laggy, but for me, they're very fast. This is the slow version. Here again, I'm not sure if you can see. I will attach those videos together with the video. But in summary, what we did was absolutely fantastic. We made a completely new model of iPSC derived airway epithelium on air-liquid interface. And we isolated the basal cells, which can expand to large numbers, therefore making a suitable model for scaling up and going high throughput. But the question still remains on how similar this model is to the already available primary models, and especially in terms of xenobiotic metabolism, enzymes, and transporters. So for that, we used TempoSeq, which I'm sure you already heard quite a lot from the previous presentations by the other ESRs. It's a directed RNA sequencing approach, and it has a library specifically designed for toxicology. And it allows comparison between our models within IN3, which is a great thing. But first, I compared the gene expression of the iPSC and the primary model together. And what we can see here is that the gene expression is very comparable. There's a line forming here with a Pearson correlation of 0.45, which is quite impressive. Moreover, in between samples, the variability is quite low, which is also quite nice. And finally, when we look at the most expressed genes in each model, we find a lot of lung genes, such as keratin 6A, keratin 17, and exin 1, mucin 1. And most genes, as you can see, are similarly expressed in both models, vice versa. And when we do a PCA analysis, we can also see that the primary model clusters with the iPSC lung airway model compared to the other IN3 models, which are endothelial, neurons, and podocytes. Then we looked at the expression of xenobiotic metabolism enzymes, which are quite important in terms of how the cells would respond when treated with drugs. Overall, here we're looking at the green, which is the primary, and the red, which is the iPSC. Overall, the iPSC seems to be slightly lower expressed than the primary, but generally, the airway model has higher expression than the iPSCs. In the primary only, we have alcohol dehydrogenases, AKR1C2, 1C3, and for the cytochrome P450 family, which is one of the main ones, we have CYP2B6 and CYP2B7P, which are not expressed by our model, which shows that there are still some limitations. However, when we look at the most expressed one, it's CYP4B1, and CYP4B1 is a lung-specific CYP, which further shows that it is a lung model that we're looking at here.